Welcome to this course, NPTEL online certification course on organic chemistry in biology and drug design. Myself, Professor Amit Bosak, Department of Chemistry and School of Bioscience, IIT Kharagpur, will be the chief instructor along with my research scholars who will be assisting this course to conduct the tutorials and to answer the queries and questions that you will have during this course and also at the end of this course. Now, let me first give you the background of this course or the genesis, that why we are trying to enter into a field which is the field of biology and also drug discovery process as an organic chemist. Now, you know, organic chemistry has started when Euler synthesized urea from ammonium cyanate, which is a landmark discovery because prior to that it was believed that organic molecules exist only in living systems. But Euler showed that an inorganic material, ammonium cyanate, could be converted into a substance which is urea that is present in the living system. So, that basically is the onset of organic chemistry. And the next 2 centuries, the organic chemistry mainly concentrated on isolation of molecules from natural sources as well as unnatural sources, and their structure elucidation, that what is their constitution. And then came the subject of stereochemistry, that means the 3-dimensional orientation of the different groups in the molecule. Along with this, the organic chemists also started to look at the reactivity of molecules, on what are the parameters this reactivity depends. So, it grew in a very rapid pace, and the another motto of organic chemistry, a big emphasis was put on how to make molecules, which is called the branch of organic synthesis. Now, today virtually we have different kinds of reactions which allow us to make extremely complicated molecules. So, organic synthesis has reached its pinnacle of success today. Now, the question is that what after this, when organic chemistry has developed so much. Now, alongside with chemistry or organic chemistry, another subject was growing parallelly, and that was the subject of biology. And the domain of biology was basically understanding the life processes, what are the different organelles, different cells, different tissues, different organs that are present in the living system, and how do they cooperate, what are their functions. But the point was that the biologists were looking at the living system at the macro level, that means what are the components and what are their functions. The more intricate details, that what are the fundamental components that are present in these compartments like the cells or the organs, or precisely what are the molecules that are present which cooperate with each other and sustain what is called life. So, it took a little bit longer time when Linus Pauling gave the structure of the complex biological molecule called the proteins. And that is basically that started the era of looking at biological system from molecular perspective. Linus Pauling precisely gave the structure of proteins, and he gave the alpha helical concept that proteins can fold in a helical manner. And then subsequently Watson and Crick, they gave their landmarks double helix structure of the double strand DNA. So that was basically the starting point of a new type of subject, when people felt that yes, we have to look at the biological system from molecular perspective. And when you talk about molecules, these molecules are present in the living system, and when these molecules are present in the living systems, that means they are basically mostly the organic compounds, that means they are carbon based compounds. Along with carbon, we know that they have hydrogen, phosphorus, nitrogen, oxygen. These are the main elements that are present. So that was the start of the looking at biological system from molecular perspective. And when you talk about molecules, then organic chemistry have a big role to play. So that's why today's chemists, the new brand of chemists, we have to generate who must know not only the basics of organic chemistry, mainly the structure, the function, the stereochemistry, the reactivity, the synthesis, the bond forming processes. They have to know all these things, but alongside we should also know that, or the new brand of chemists, they should also know how biological processes take place in the living system. Remember, interestingly, these reactions which are taking place in biology, or the molecules which are there present in biological system, in a living system, they are not different, because they are basically organic molecules. And the chemistry that takes place also, they are nothing but organic chemistry, organic reaction pathways that are taking place. Only difference is the kind of environment that is present in biology and when we do organic chemistry in the laboratory. That is the basic difference. We do organic reactions in test tubes or in flasks, and so that is a very isolated system. But the biological system, the biological reactions take place take place alongside with the surroundings. That means so many molecules, so many catalysts, so many macromolecules are present in the system, living system, but still we can have only one reaction in presence of so many reactive molecules. Now, these reactions are done by what we can call the carpenters of biology. That means they do the synthesis, or they do the breakdown of big molecules. So they are basically the biological carpenters, and they work in perfect harmony. If the harmony, that means harmony means one after another, it is a harmonized process, and once the harmony breaks down, then what happens? That we suffer from lot of problems, what are called the diseases. So the diseases are basically related to this harmony that takes place in the biological system. Okay, so I think now it is amply clear that today the chemists have to know the basics of the basics of biology and what type of chemistry that goes on in the biological system. At the same time, the biologists now should venture around, they have already done that, that they should go or they should look at the biological system from the standpoint of molecules, that what are the exact microscopic details that take place when something is synthesized or when something is broken down or when a signal has to pass from one cell to the another. Okay. So, that is basically the genesis of this course. So, I realized that this type of course is extremely essential for today's chemists and biologists. Just one point to add is that right in the around 1990s, world renowned universities, they have changed the name of their department. From Department of Chemistry, Harvard University named their department as Department of Chemistry and Chemical Biology. So, they realized at that time that this new brand of chemists should be called as chemical biologists. Okay. I think that is the that justifies the that why this type of course will be important, and I will try my level best to show you organic chemistry in biological systems. And then after we do the biological system study from organic chemistry point of view, that will generate a platform from where we can utilize our knowledge and then show that how different drugs have been designed, have been made, and then now they are extremely, we know that they are useful. And so, you have a clear perspective of the whole process, that from chemistry to organic chemistry, and then organic chemistry to biological processes, and from biological processes we will go to the drug design. So, there is a chain of events that will happen in this course during the deliberation of this course. Now, let us talk about the biological system. As I said, we will look at from the molecular perspective. Now, biological molecules, or the molecules which are also called the molecules of life, mainly comprise of 4 types. These 4 types of molecules, they are present in all living systems, with few exceptions possibly. Okay. Now, these 4 different types of molecules are proteins, the number 1, then the nucleic acids, lipids and carbohydrates. These are the 4 major components. Now, most of these are called biological macromolecules. However, I can say that macromolecules are basically made of small molecules. So, small molecules join and finally give the macromolecules. So, obviously, when we talk about these macromolecules, we should talk about the building blocks of these systems. Now, all are not macromolecules per se. Like if I talk about lipids, the lipids are basically molecules which are molecular weight less than say 1000. So, they cannot be macromolecules. So, but proteins, nucleic acids are macromolecules, and carbohydrates are basically they could be small carbohydrates like glucose or fructose or sucrose, all these things, or they could be large branch of carbohydrate molecules tied together like what is present in starch or cellulose. Now, apart from these 4 basic classes of molecules, the proteins, the nucleic acids, carbohydrates and lipids, there are other molecules. They are small, they could be small molecules, that they could be medium sized molecules. They are also present in biological system, and their role is also extremely important in in coordination, in maintaining the coordination between the cells. Okay. Coordination means it is called a process which is signal transduction. That means one cell gives a signal, that signal goes to another cell. Now, who actually creates these signals and also transmits these signals? So, in that case there are several small molecules which play a role, but these small molecules are mainly generated from some of the other molecules which are present in the biological system. Now, when we say that a small molecule is generated in a biological system, we have a specific term for that process, that is called biosynthesis of molecules. Biosynthesis is nothing but synthesis, but synthesis taking place in a biological system, and the term synthesis is specifically reserved for organic chemists to do their reactions. Okay. So, we will cover all the aspects, all these 4 class of molecules along with several biomolecules which have small molecular weight. We will do that. Okay. Now, let us come to the building blocks of the molecules of life. Today it will be just the preliminary aspect of all these molecules, and then we will go deeper and deeper on the structure and function of these molecules. Now, these molecules of life, the first one I told you about the proteins. Now, proteins, you already know what are proteins. We know that their basic building blocks are amino acids. Okay. Now, these amino acids can be of various types. When we say amino acids basically compounds where there is an amine and a carboxylic acid group. But these amino acids which are present in the biological system to make the proteins, they have a special character. They are called alpha amino acids. Okay. Alpha amino acids means the amine is present alpha to the carboxy group. Basically, we have the molecules of life, and these are, you have proteins, nucleic acids, carbohydrates, and lipids. But remember, these are the large macromolecules. Alongside, we have small molecules and also medium sized molecules. These are also extremely important. Although these 4 are classified as the molecules of life, but it will be very interesting to know that some of these small molecules are also extremely essential to maintain the life processes. Now, we were talking about the building blocks of these large molecules. Okay. And the first one was, we took up your proteins. Now, we know that the proteins are made up of, if you say only amino acids, that is not enough. You have to say that they are made up of alpha amino acids. Okay. Again, that is not sufficient. Alpha amino acids, I can draw the structure, is basically a R and then NH2 and CO2H. Why it is alpha? Because this carbon is the alpha carbon. Okay. So, alpha amino acids. However, if you look at this molecule, you see that there is a center, there is a hydrogen here also. So, this carbon is what is called a stereogenic center or the classical a classical nomenclature is a chiral center. Now, so that means this can exist in two forms, two enantiomeric forms, and interestingly in all alpha amino acids which are present in the living system to make the proteins, they belong to the class of what is called L amino acids. So, they have a configuration which is L amino acid. So, what are the components of the proteins or building blocks? You have to say that they are made up of L alpha amino acids. Okay. Now, there are you possibly know that the number of amino acids that are present in proteins are 20 alpha amino acids we know. Now, except glycine, where R is equal to H, that is your glycine. This glycine, because of the lack of R means some alkyl or aryl groups, what you have is hydrogen. So, that makes it a non-stereogenic carbon, and if it is non-stereogenic, so there is no question of stereochemistry here. So, glycine is the only achiral amino acid that is present in proteins. All other amino acids are chiral and they have the L configuration. Now, this L or D, this DL nomenclature for assigning absolute configuration is little bit, it is an older version of telling the absolute configuration. Now, this has been replaced by RS nomenclature in organic chemistry. However, biologists still prefer this DL nomenclature in organic to say the absolute configuration of the biomolecules which are chiral. Okay. Now, this L configuration can be translated into RS nomenclature, and if you do that, you will find that these 19 amino acids which are chiral, these 19 amino acids except cysteine, there is one amino acid which is a sulfur containing amino acid, except cysteine, all other amino acids are having S configuration. So, L amino acid means S amino acid, but remember except cysteine, and glycine does not have any does not have any chiral center. So, this is the building block of proteins, alpha amino acids. Now, because it contains, we know that the amine is basic functionality and carboxy is an acid functionality. So, in a molecule you have both acid and base present. So, that presents an interesting situation. So, there can be internal hydrogen transfer between that, and so NH2 can be present as NH3 plus and CO2H will be present as CO2 minus. Okay. So, now if you take an amino acid and put it in an acidic medium, suppose a pH say 1 in pH 1 aqueous solution, then what we have, then the NH2 will basically be protonated, because this is an amine, and that will be protonated, and so that will be NH3 plus. So, if an amino acid is taken in an acidic medium, suppose which has got a pH of 1, then what will happen, this NH2 group being a basic group, that will be present as NH3 plus. The carboxylic acid will be present as CO2H, because at this pH the carboxylate ion cannot be present, okay, because carboxylic acid has its own pKa value, means the own pKa means the pH in which the dissociation takes place. So, below the pKa it will be present as CO2H, and then suppose you have this R group. So, at pH, acidic pH, this is the situation, and as you raise the pH, that means you are adding alkali. So, what will happen? So, now the question is, you have two acidic hydrogen, this is the CO2H, the hydrogen is acidic, and this is NH3 plus, this hydrogen is also acidic. So, they one of these will first come out, and obviously this CO2H has a lower pKa value. So, that will come out, that will be abstracted by the base, and so that will be present as CO2 minus, and this will be the structure of the amino acid. So, now you have a molecule where there is plus and minus both are present. Now, this is what is called zwitterion. So, that is the zwitterion structure of amino acid. Remember, this zwitterion will be present at a particular pH, and then if you increase the pH further, what will happen? Next proton that can be abstracted is the from the NH3 plus. So, that will be abstracted, and in the process you will get the NH2. Now, that the hydrogen has been abstracted by the base, and that will be CO2 minus. So, this is the end. After that you do not have any hydrogen to pick up. There is a hydrogen here, but its pK is much way above, which cannot be abstracted by a simple base as such. Okay. So, these are the two ionizable hydrogens that are present in the system. Now, this dissociation from the protonated form, or the total charge of the system is +1. Here the total charge of the system is 0, and here the total charge of the system is -1. So, it is fluctuating from +1 to -1, but going via a zwitterion which has got a 0 no net charge. Okay. Now, this is the situation in case of amino acid if you start titrating it from pH 1. Now, at some pH what will happen? It will be present in the form, in the zwitterion form, and at that point or at that pH the whole system is electrically neutral. So, if you want to put this amino acid suppose in a gel, and then you try to start applying a voltage, a plus on one side and minus on one side, and put the amino acid say in the middle, then what will happen? Depending on the charge, whether it is a negative charge or positive charge, it will start moving in one direction or the other. But if it is present in the zwitterion form, then what will happen? It will not move, it will stay there. Now, what will be the pH at which the zwitterion is present? That pH is basically, we can deduce it later on. That pH is there is a formula there. The pH is basically Pi. This is what is called isoelectric point. Pi becomes half of pK1 plus pK2. What is pK1? pK1 is this one, the first dissociation. This is K1, this is K2. Remember, these are not rate constants, these are equilibrium constant between these two species. So, that is the general relation of Pi. Remember, what is Pi? Pi is called isoelectric point. Isoelectric point is nothing but, remember, this is not a point, this is a pH. The pH at which it is electrically neutral, it does not move on any side when you apply voltage on this molecule, which is now applied on a matrix in which it can move. Okay. Now, this is a very important parameter of amino acids. Here I have taken the amino acid where there is only the alpha amine present and the carboxy present. Now, what about this R? Depending on this R, we have different classes of amino acids. Okay. And if the R, what type of different R's are present in amino acids? The R can be very uncharged, a non-polar group, and these amino acids will be called non-polar amino acids. So, the amino acids are classified according to the nature of the R. Some amino acids are called non-polar amino acids, and that means the R is non-polar, say a methyl, which is alanine, and if it is isopropyl, that is called valine. If it is CH2PH, that is called phenylalanine. So, different types are there, but they are all non-polar amino acids. Then that means this side chain is such that they does not like water to be surrounding them. Okay. So, they are hydrophobic in nature. If R is a group which is polar group, but not charged, polar group means that there is a dipole moment present there, and then that can form hydrogen bonds with surrounding molecules which can have donor hydrogens present. And these R could be CH2OH having a hydroxy group, the R could be CH2SH having a SH group, or it could be CH3OH, that is what is called threonine, and these OH groups or SH groups have the ability to participate. First of all, they are polar groups, because we know that sulfur, oxygen, they are more electronegative than carbon. So, that generates a polarity, and this generation of polarity creates, or it gives a ability of the atoms like oxygen or the sulfur to participate in hydrogen bonds. So, they are the amino acids with polar groups, but uncharged. Then you have amino acids where this R group contains another acidic group, and like you have NH3 plus, say CO2 minus. I am talking about just writing the deuterionic form, and you can have another acid group, a CO2H group. So, an extra carboxy group. Now, this particular amino acid has 1, 2, 3, 4, 5 carbons. So, that is called glutamic acid. And so, they are amino acids containing acidic functionality. Okay. Acidic functionality means in the side chain, that is called the side chain of the amino acid, and you can have slightly one carbon less also, the lower homologue of glutamic acid, which is called aspartic acid. So, these are the acidic amino acids. Then you have just the reverse, the basic amino acids, where instead of carboxy group you have other different amino acids which contain either NH2, or it could be which is called imidazole group. Imidazole group is a functionality, it is this is what is the imidazole functionality. So, this is a basic group. So, now depending on whether there is NH2 present or imidazole present, or it could be arginine, that is another functionality, arginine group, arginine that has called a basic basic group. So, these are called the basic amino acids. So, the basic amino acids are mainly lysine and arginine. There is another amino acid which is called histidine, which is much less basic than arginine or lysine. So, the strongest basic amino acids are your lysine and histidine. So, these classifications are there for amino acids. So, now what we have learned, that first of all there are 4 building blocks of the life, which are called the proteins, the nucleic acids, the carbohydrates and the lipids. And then we said that alongside these large molecules, or these 4 major class of molecules, you have small molecules which are also extremely important, and they are generated inside the body, biosynthesized, or some other are taken also. I forgot to mention that some of these molecules come from our diet. They are extremely essential again to maintain the life. And then we talked about the building blocks, the monomeric systems which makes these biological macromolecules like proteins or nucleic acids or carbohydrates. We have only talked about proteins. Proteins are made up of combination of L alpha amino acids, except glycine. Glycine does not have a chirality, okay, and they join together through bonds, which we will talk about. They are joined together by amide bonds and then give different varieties. You have 20 amino acids. So, there are different permutation combinations you can make, and that will make all these different diverse array of proteins that are present in the body, and they have different functions. Some proteins could be have catalytic power, and they are called enzymes. Some proteins could be structural, like the collagen, which maintains the muscles, or some proteins can be hormones also, like insulin. So, there are different, or some proteins could be other structural, like they are present in the membrane surrounding the different cell contents. So, but that will come later, the functions. First, let us focus on the structure of this. So, this is the first one. The most important point of amino acids is the isoelectric point, because isoelectric point will ultimately decide the charge. When different amino acids are present, what will be the charge of a protein at a certain pH, that can be calculated only on the basis of the isoelectric point. What is the definition? The definition is isoelectric point is the pH when the amino acids are electrically neutral and do not move in an electric field. Okay. So, this is the first half of the lecture on amino acids. We will talk about the other issues of amino acids in the second lecture. Thank you.